Good day to all viewers. My name is Mukesh Ramdani. I'm an Agricultural Officer 1 attached to the Entomology Unit Research Division. My presentation for today would be on plant diagnostic services, insect pests and diseases. So I'd like everyone to sit back, relax while you're all having lunch, or probably may have just had lunch, and I'll just try to be not too long, right? And at the end of the presentation, see if I can answer most of the questions that you all might be posing to me. So I want to get into the slide presentation for today. And at the research division, the crop protection subdivision comprises of two main units for plant pest and disease diagnosis: the entomology unit and the pathology unit. The entomology unit has 3 agricultural entomologists, and the pathology unit has 3 plant pathologists. Each is in charge of pests and diseases of different commodities, which are divided into 3 crops: vegetables, root crops, herbs, and spices. The plant pest disease services are available to other divisions: the plant quarantine division, the demonstration stations, agri-services division, the farming communities, and the general public, which of course is at no cost to anyone who would be seeking our services. What is the importance of plant pest and disease diagnostics? Now, we can answer this in 2 ways. We have diagnosis in agriculture, for instance. Now, diagnosis is the process of determining the cause of a problem. It can be a long or short process depending on one's ability, experience, and the nature of the problem. Once the cause is known, an appropriate management strategy can be developed. And this strategy would then be given to the stakeholders, which might either be a farmer, it could be a householder, where they would be able to address the problem that is occurring. Without proper identification of the plant pest or disease, the causative agent, control measures can be a waste of time and money and can lead to further plant losses. So therefore, proper diagnosis is vital in agriculture. How are plant samples received by the units? Well, there are several ways. A farmer can request via the county offices, where he would actually go to the county offices, and there he would meet with his extension officer and request for a visit to come if he is getting a problem with his particular crop that he's planting. And there, right there and then, the officer himself would either visit and take samples and answer the questions to his best of his ability, or if not, then he will have to bring it to the research division. The request can also be via phone calls directly to the division, where we, at the end of the presentation, we have an email address where you'll be able to send like photos, right? The farmers or any of the stakeholders, householders as well, you can come into the research division. And we also have interception by plant quarantine officers. This would be for people who would be bringing in commodities that are imported. All right. So it's a very, very important task that we have to do. And both units, the entomology and pathology units, are there to help. Now, diagnostics can be in the field or at the laboratory. Now, in this slide, would show a plant with a bug on it there, and that would be probably would have been brought in by some farmer, and that is where some sort of problem would have been occurring, and the entomologist at hand would be looking at it in the lab to try and probably give the best answer as best as possible. That would be to the side to the left, and then to the right, the picture to the right is where you would have the entomologists and other officers from research division visiting a field, and at that point in time, based on the symptoms and signs that you'll be seeing out in the field, possible field diagnosis might be given to the farmer together with a strategy to address the problem. Now, in terms of the field diagnostics, this is based on past and present experiences by the officers who could give a preliminary diagnosis of an insect, pest, or disease, and it can be done in the field. Now, the parts of the plant affected are examined for the presence of the insect or disease symptoms present. A positive identification of the problem can be given based on the officer's experience. And then some sort of management strategy would then be provided as a solution for the particular problem. In the next slide, the other slide shows an officer in the field there where the officer is actually talking to the farmer and showing, right, what are the possible signs and symptoms of the disease, and that is the picture to the left. And then the one to the top right hand corner, that is what would have been the actual tree, the tree itself that was affected. Right now, this is from all experiences, this tree would have been or succumb to red ring disease by a vector called the Rhynchophorus palmarum. And in terms of confirmation in the field, what will happen is that the officer would get assistance in cutting the tree, and in cutting the tree, if you see in the red ring band around it, that will be a field confirmation of the presence of the disease. And at that point in time, the officer will be able to then give some sort of recommendation, which will be in the next slide. And in this other slide, it shows a pheromone funnel trap, which would attract the vector, which is Rhynchophorus palmarum. And the vector is at the bottom. You would see it enclosed probably within a glass aquarium, right? And those are the vectors that tend to transmit the red ring disease, which is of course is caused by a nematode. And last week, one of the presenters, Mr. Vidal Balraj Singh, would have gone into controlling and recognizing this red ring disease. And he would have also given you all an example as well of the funnel trap, which works very well as a management strategy in controlling and managing this disease. Right, so this next slide the other slide shows a farmer with problems with his melongene, right? And the officer would then look at the leaves, examine the under surface of the leaves, and right there and then he would be seeing problems with at the lower left-hand corner, you probably might be able to see some white specks, and that would be white flies in the field. That would be causing the problems that the farmer would have. So right there and then, again, the officer would be able to give some sort of recommendation to address this problem. And to the right, left-hand corner at the bottom, right, the farmer would have had this problem in the field for a period of time where it would have been the pigeon pea borer, but the pigeon pea borer is something that you wouldn't control in the late stages of a crop because the crop would have already been going down, and you would want to recommend something for the farmer when he plants his next crop for the next time, so that he'll be able to probably get a longer bearing of the trees and harvest a lot more pigeon peas, which would not be affected by the pigeon pea borer. How a plant sample should be collected for diagnosis. Now, if the officer is unable to identify the problem in the field, then a sample will have to be taken from the plant. It could be a part of the plant, a whole plant, or a fruit. And then that sample would then be collected and brought to the laboratory. But it has to be taken in a particular manner for it to reach the laboratory in such a state that the particular pest or disease would be able to be identified. Now, samples that are collected would be based on the size of the plant and, as I mentioned before, the parts of the plant that are affected. So you would want to use a clean, clear plastic bag to put the sample in, and that will be properly labeled with at least the date, right, the name of the farmer, and that should be the address of the farmer and possibly the crop that the farmer is producing. The samples then, as I mentioned, could either be the stem, the fruit, the leaves, or the roots. And that root could either be taken with soil or without soil. The sample bag should then be properly sealed. That sealing could either be tying or stapling to the top of the bag so that you wouldn't have any of the particular agents escaping. And then the samples would then be received from the farmers or possibly general public, or even by other officers from the field who would be bringing in the samples, right? They would bring it to the research division where it would be received and then taken for processing. Now, the other side would show a possible way one of the sample can be taken from an affected plant, and it would just be part of a plant. And here we have the officer taking the sample from part of the plant. It will be a leaf sample, so the plastic bag is actually inserted over the part of the plant. It's removed with a secateur, and then it will be gently tied and placed in, if you have a cooler at hand, depending on the temperature, so that the parts of the plant, it would reach in a proper condition to the research division. Now, when the sample reaches the research division, a member of staff would then take the information and record it on a sheet. The sheet is seen on the right-hand side of the slide, and all the information is recorded there for reference and also for contacting the farmer at a later period in time. So then the staff would either inform the entomologist or the pathologist who will then look at the sample. So from here now, we're just going to a little briefing on the methods of the diagnosing in the laboratory, right? There are different methods of laboratory diagnosis. So some people tend to use what we call the picture book method, and this is where the problem is matched with pictures from a book, a reference book, to help diagnose the problem. Then there is the use of what we will call diagnostic keys or taxonomic keys, right? In which insect identification can be done, but this is a long and timely method. But it's the most accurate method of identifying the causative agent. And then within the past year or so, we have been collaborating with CPDN, which is the Caribbean Pests and Disease Network, where we would be taking pictures of pests, particular pests and diseases which we wouldn't be able to identify, or at least which we are unsure of, and it would be taken and sent via email for identification. And so far, it has been working great in terms of one of the diagnostic tools that would aid the entomologists in identifying particular pests. Now, this other slide shows some of the pictures that are taken, right, when the samples are brought to the laboratory. In the left top, you would see like the barnacle scales, right, and the right top is where you would have the particular officer or entomologist looking at the pest under the microscope and trying to use the picture book method to identify the pest to develop some sort of positive identification. In the middle, you would be seeing a banana, middle bottom, with a mealybug, and that would have been an interception by plant quarantine officers. And right there and then, we would have to try as best as possible to give some sort of tentative identification, because this interception would have been a commodity that would have been something where an importer would have brought in the bananas and it would be held. All right. So something quick has to be done, and a possible ID would then be given, and then a strategy in terms of probably most likely fumigation of the pests or fumigation of the container in order to get rid of the pests would be done, so that the particular commodity can then be released to the importer. This other slide shows pests again of probably interception by plant quarantine officers, and you would notice that at the top right hand, right there, you would be taking out the head of the pest to show the particular structure of the head, the shape of the head, and then the antennae. So you'll be checking the segments of the antennae, right, where you will want to match that up to at least probably the family, and maybe come down to at least the genus of that particular organism, so that you'll be given a strategy to at least manage that pest. And if not, and you cannot give an identification, these pictures would then be sent through CPDN, where we would be able to either be given some sort of tentative ID based on it. Now, if it is a pest that is not here and is not present within the country, then that pest would have to then go through several process where it would have to be shipped for ID, and we'll have to get two authors in terms of identifying that problem. And then now that problem of the pest being in the country, then that will have to go through a whole process of being reported to IPPC, which is an International Plant Protection Committee, so that everyone would know that that particular pest is now present in Trinidad and Tobago. Now, in some instances, samples are brought in, and those samples that are brought in could be in the form of most likely possibly either the egg stages or the larval stages of the insect. And in the entomology unit, what we'll have to do is then take those samples, place it on some sterilized sand, and those, if it's larvas at the larval stage, we'll have to continue feeding those larvae until it begins to pupate. Now, in this slide, you will see some pupa, and I think this would have been of the tobacco hornworm that would have been brought in, and it would have reached the pupal stage there, and then from there, we would leave it until it emerges into the adult, and then from there, the adult stage, you'll be able to more or less positively identify the particular pest. So we would have talked a little bit lengthy on entomology and bringing in samples to the entomology unit. Now, I'd just like to talk briefly on the process of diagnosing diseases in the laboratory. Now, we tend to go through three things here in terms of receiving the sample, preparation of the sample, and actually incubating the sample and isolating it for identification. On the same light, the samples are received, and when the samples are received, they are taken to the pathology unit, and there is a receiving room where all the information is taken by someone, a member of the staff, and placed on a form. Now, at the left-hand bottom of the slide, the pathologist might, if it suspects that it might be something like bacteria, they could do a simple test as the bacterial streaming, where they will take a part of the stem and immerse it in water in a petri dish and to look for streaming. And if streaming occurs, then that is a tentative identification of a possible bacterial problem within that is causing the plant to be affected. If the problem cannot be easily diagnosed, then what will happen is that the sample will then be further taken, and the organism goes through a series of inoculation for isolation, and then placed on slides, and then after it is placed on the slide, a dye would have to be placed on it for viewing under the microscope, so that the particular organism would be able to be identified. And here we have a member of the staff looking at the slides which are already stained, right, for a particular organism, for a disease. And then on the other slide there, you would probably see on the right hand bottom of the slide, that's a nematode, which would have been picked up whilst doing the various isolation. Now, some pathogens tend to have specific symptoms, and also it will tend to give certain shapes on the plant part in which it is attached to. And this can be used to give some sort of preliminary diagnosis for treating. Now, if this can be done, then the pathologist can then give some sort of management strategy to the farmer to manage this problem in the field. If not, well, then the process of slide making for pathological purposes tends to be a little bit lengthy, and once it's done and the organism is isolated, the farmer can be given then a better strategy, management strategy, to more or less manage what the problem in his field. Now, the other slide just goes into a little bit of the same thing with the causal agent of the problem, where samples will be taken and viewed under a microscope. One of the things that the entomology and pathology unit is that all samples that are brought in are dealt with, and we try to give a timeline in terms of actually dealing with these samples. And to the best of the ability of the pathologists and entomologists, what they would tend to do is try and develop some sort of management strategy so that the farmer would be able to address this problem as soon as possible. Now, if the field is an old field, well, then the farmer will have to look at addressing the problem if it occurs when he plants again, if it's the same crop, right? Or a crop of probably maybe within the same family, and if the same problem occurs. What I would like to reiterate here is that at the crop protection subdivision, all the services provided by the entomology and pathology units are free and can be accessed by all the stakeholders in agriculture. So what you can do at the end, the next slide will show that you can contact us via email with ento.diaglaboratory@gov.tt, and I think that would have been the email used by all the other presenters at the end of their presentation. Right. So you would see it showing up in the slide. Right, so you'll see the email address showing up at the bottom of the slide there. Ento, ENTODIAG, laboratory is a capital L, LABORATORY @gov.tt. You can upload digital photos, or you can leave a contact number where we can reach you at, and we would call as soon as possible or if you uploaded your digital photos, we will try as best as possible to try and give you a tentative diagnosis of the problem. And if not, we will then have to probably do a field visitation to probably get more samples or try and see if we could do a diagnosis at the field, or then bringing samples into the lab to try to see how best we can give you a problem solution to your problem. This here, the last slide, shows some of our dedicated entomology staff members. And the previous slide before you would have seen some of the pathology staff members. But these entomology staff members, they are very dedicated to their work. And all of us at the crop protection unit try as best as possible to try and help any member of the public, or it could be a staff farmer who would come in to receive services at the crop protection subdivision. What I'd like to do at this point in time, I'd just like to say thank you to my colleagues who would have assisted me today, Mr. Mara Ganai Singh and Mr. Sunil Ramnarayan, and also the Deputy Director Crop Protection for giving me the opportunity to present on this topic. I'd also like to thank all the viewers for logging in and their patience, and the personnel at the communication unit who would have done the live stream, which you all have viewed today. Thanks again and have a safe day. Any questions, you all would be able to type it in, and we'll try as best as possible to try and answer the questions now.
Look at you.
Type of fungus.
Okay, well, just say no.
Yes. Okay, this one.
We'll read it out and answer it.
Fine.
So one question.
Okay, we have a question here from Mr. Clive Spencer. One of the first questions was that, is it true to say that hydroponics has less pests and funguses than if you are planting in soil? The answer to that is yes, because what happens is that the environment, which would control your biotic and abiotic factors, it is better controlled than having been planted in soil media. And most of the times in the hydroponic system is above the ground, and the amount of nutrients that is given to the plant is in such a way that the plant is healthier and it can survive any attack by probably most of the pathogens. Now, if an insect pest do come in, it is better being able to control, because you can see the problem being affected almost immediately, and it can be addressed as such. Now, Mr. Brian Ramlakan, you pose a question here on the curling of pepper leaves. Now, the curling of pepper leaves, that's kind of tough, because you can have a lot of causative agents that could cause the curling of pepper leaves. You see, you may have a viral problem. You could have problem with insects, and those insects could range from mealybugs, thrips, mites, right, that could cause the curling of the pepper leaves, right? And then what might happen is that we will want to say that we recommend something on the curling of the pepper leaves. If you probably could take a photo and send it to us, or even contact us and send a contact number, we'll be able to contact you and probably do a field visit. Now, another question here from Mr. Clive Spencer. What is a good all-round insecticide to use for grasshoppers, moths, whiteflies, aphids, loopers, bush bugs, etc.? Right. Well, that's a good one. Now, most of the times is that if you want to control all those bugs at the same time, you would want to use something that is systemic. Right? And in using a systemic insecticide, you have to be very careful too, because those systemic insecticides tend to go through the plant system. And if it is that the plant is already fruiting, right, that insecticide could go to the fruits. Now, there are insecticides that are systemic and safe, but those insecticides, they tend to be a little bit expensive, and the post-harvest interval tends to be a shorter period of time. Now, offhand, I do not want to give the names of insecticides, but I probably could give you the active ingredients, and you probably could look it up. There is one where the active ingredient is pyrotetramat. All right. There is another insecticide where the active ingredient is acetamiprid, and that has something called translaminar action, where when it is applied to the plant, it actually goes through the leaves of the plant, so it cannot be applied to the soil, so it's only absorbed through the leaves. And there is another one by Avion Subrati. If a tomato plant is showing signs of tomato yellowing, leaf curl virus, are there any sprays, etc., that can be used to help the plant recover, or should it be removed and destroyed? Well, you answered your question there in the last part. In most instances where you have that occurring, you would want to remove the plant and destroy it, and you would want to also look at your other plants which are healthy. If you do not have an insect problem like whiteflies, because whiteflies tend to be the vector for the spread of the particular pathogen. So what you will have to do now is look at your plants and see if you have whiteflies underneath the leaves, and you would have to then come in and use same acetamiprid, which has translaminar action, or this pyrotetramat to control the whitefly problem. And once you control the whiteflies, well, then you will be able to manage, right, your tomato leaf curl virus. Wendy Ramkisoon, what causes brown tips in avocado trees? Well, that's a tough one, because avocado trees can be affected by several things. Right now, with the dry season, you will have a lot of problems with what we call the avocado lace bug, and you have to turn the leaves to the underside and see first of all if you have an insect problem underneath there. The avocado lace bug is it has a very shiny wing pattern to it, and the immatures tend to look like almost like if it's black pepper spots at the bottom of the leaves, and you would tend to get the browning of the leaves occurring, leaf fall. Another problem there is that if you have water stress in avocado would start from the tip of the leaves coming inward. Right, so you need to look at the various factors affecting the avocado, because with the dry season, which we are having a more or less an extended dry season, you will tend to find that you have a higher incidence of pest problems. Now, with the onset of the rainy season, you will tend to have a higher incidence of disease problems. All right, so we need to look at the environmental factors. Right, from Councillor Ramona Victor for Siparia East, what causes pepper to get black and spotted without ripening? Right. Now, like I said before, you could have problems with a disease, something called anthracnose. That's one of the first things that might be able to come to mind that you might have, where the pepper would be able to would be unable to develop into a ripe fruit once the pathogen attacks early. Another thing is, if you have uneven watering and less calcium in the soil, you can get a physiological problem of the pepper, right, which almost occurs in tomatoes, and you will get it in peppers occurring as well too. Blossom end rot, that is what would be one of the physiological problems. You could get sun scald with the weather conditions we have in right now, and the fruit itself would not develop and go to the ripening stage. Spell
Spell the spirotetramat.
spirotetramat.
That's in my bird. I was going to spell it.
Yeah, spell it. Yeah, go ahead. SPIR. Yeah, that's correct. TTR.
Oh, you want to see it?
Now let's see it. All right, spirotetramat. Mr. Kleispander wants to know. Spirotetramat is SPIROTETRAMAT. All right, you can Google it, and in Googling it, you'll be able to see the trade names and also what particular pests it can be used for. Asher Lee planted 3 okra seeds, of which only 4 germinated. One of the plants was growing very healthy and with what looks like 6 small okras at the top. It did not flower. Over the space of one day, the top of the main stem just bent down 180 degrees with the top leaves. Right? The leaves were so bent that after about 2 weeks, the leaves started turning yellowish and were contracting inwards. Okay, most likely what you probably would have had there might have been a mealybug problem. But you see, in terms of observing your plants, right, you started observing when the symptoms probably would have been almost far gone into the plant, and the tip of the okra leaves, which is the bearing part of the plant, that is where the mealybug tends to come on and affect the plant part itself. So you started getting some sort of curling. It might look like if it was initiating flowers, and then all of a sudden, as you would see, you will get that part turning yellow and start contracting inwards. But it's really a rosetting and curling of the plant that would have been occurring. Now, I am not seeing the plant, but based on what you are describing there, but if you had a picture of it, right, it would have been better to try and diagnose the problem. But from what I'm seeing here in terms of your comments, most likely might have been a mealybug infestation. And you will need to observe your plants very carefully in future, where you might be able to use a simple thing like neem or bioneem and soap water, which could be sprayed onto the plant, such as okra, every 3 to 4 days, probably about 2 or 3 applications, and that would more or less subside and take care of the problem. Even using neem. Oh, why is my right? Dominique, Nikki, John Roberts. Why is my pawpaw rotten before it's fully ripe? Several factors can result into what we call rotting of the fruit before ripening. First of all, you can have anthracnose, which is a fungal problem, right? And this anthracnose, if it's not detected early, what will happen is that you can pick the fruit, put it to ripe, and instead of the fruit ripen, the fungus will take over the entire fruit, and it will start rotting. Our next thing would be if you have any deficiency, right, within, because pawpaw in itself is a very sensitive fruit, and you can have deficiencies occurring, and that would prevent development of the fruit to go to the ripening stage, and you will get more or less the pawpaw rotting before it even start to ripe. Right. What causes yellowing? Miss Patrice King, what causes yellowing of pawpaw tree leaves and stunting? And is it necessary to plant multiple trees together for it to bear? Excellent question. Now, first of all, pawpaw tree, if you plant pawpaw trees from seed, which you probably might have bought in the market, you wouldn't be too sure whether you'll be getting male or female trees coming out from the nurseries. Now, if you buy pawpaw trees from a nursery shed, most of the times that those trees you will get would be female trees, and those will go straight to bearing, because the seeds that are selected for planting are seeds that are from that particular part of the fruit, right, where you will get female plants and not male plants. All right, and what will happen is that it is always recommended to plant several pawpaw trees and not one tree, probably 3 or 4. All right. The other part of the question in terms of preventing the yellowing of the pawpaw tree is that there is a particular disease that is spread by a vector, which is a leafhopper, and we need to control the leafhopper first, where you will have to do some sort of spraying. So you'll have to use some sort of systemic insecticide to spray your pawpaw tree as it develops. And this would have to be something that is routine, probably every 2 to 3 weeks. And to prevent it from getting what we will call bunchy top disease, right? And this bunchy top disease, right, will destroy your entire tree. Ms. Avian Subrati, earlier in presentation, you said I would have mentioned and spoke about the pigeon pea borer. Would applications of products containing spirotetramat help for this? And if so, at what stages should it be administered to prevent it from occurring? All right. Now, spirotetramat is specific to sucking insects, right? And not necessarily it would prevent larvae from developing into the pupal and adult stage, especially if the larva is enclosed within like a pigeon pea pod. Now, most of the times, what will happen is that in trying to prevent the pigeon pea borer from affecting your plant, you would have to probably do application of probably contact insecticides where Bacillus thuringiensis, BT, any BT, which is a biological, because you would want to prevent, or you would want to keep it as safe as possible, because you will be harvesting your pigeon peas over a period of time, and that might be 2 or 3 times a week. And BT is very safe. So BT is something that you could use, right? And you could use that throughout your cropping period, and that will assist. Now, Miss Patrice King, orange tree keeps flowering but not reaching the bearing stage. Flowers are falling off. What can be used to keep the flowers to bear fruit? Right. Now, citrus tend to be very sensitive to irrigation, nutrition, lack of nutrition, right? And perhaps your soil is probably lacking either calcium, or the pH of the soil is probably it's a little bit too acidic, so that even if you are applying nutrients to the soil, the plant itself is not able to take it up. So you may need to probably do a soil test so that we can determine what the pH of the soil is, so that if the pH of if it's too acidic, then we'll be able to tell you you might have to use some limestone to neutralize the soil, so that it will be the plant itself would better be able to absorb the nutrients, so that the flowers would then be able to go to the fruiting stage without falling off. I'd like to thank everyone for their questions, and if there are any more questions, that would be answered later on, right, through through the email. So thank you all very much. It was nice, and have a safe and pleasant day. Thank you all. Thanks again.